I am a bull shark, Carcharhinus lucas, a predator that defies the boundaries most of my kind respect. While my relatives patrol the open ocean, I slip between salt water and fresh, my body a testament to an adaptation so rare among sharks that scientists have spent decades unraveling its mechanisms. My family, the Carcharhinidae, includes more than 50 species of requiem sharks, but only a handful can tolerate even brackish water for brief periods. I go further. I travel up rivers, sometimes hundreds of kilometers from the sea, entering lakes and tributaries where no shark should survive. My scientific name reflects this duality. Lucas comes from the Greek word for white, describing the pale underside that countershades my body. Carcharhinus marks me as a requiem shark, a group named either for the funeral mass, requiem, because early fishermen considered us dangerous, or from the French word requin, simply meaning shark. The taxonomy places me in the order Carcharhiniformes, the ground sharks, which account for the majority of shark species alive today. Within this order, my family stands out for sleek, streamlined bodies built for speed and endurance. But within my family, I stand out for something else entirely. My kidneys, my gills, and my rectal gland work in concert to regulate salt in a way that lets me survive where almost no other shark can. I am built heavy and muscular, my body more robust than the streamlined silhouettes of blue sharks or the elegant curves of blacktip sharks. Adult females like my mother reach lengths of 240 to 340 centimeters, measured from the tip of the snout to the end of the upper tail lobe. Males grow smaller, typically reaching 200 to 290 centimeters. Weight varies with condition and reproductive state, but a large female can weigh 300 kilograms or more. My body is stocky, my girth substantial, my head broad and blunt. The snout is short and rounded, not pointed like a spinner shark's or flattened like a hammerhead's. This gives my face a pugnacious look that humans often describe as menacing, though to me it is simply the shape that houses my sensory organs in their most efficient arrangement. My coloration is a study in countershading, a pattern shared by countless marine predators. My dorsal surface is gray, sometimes with a brownish or olive tint, a shade that blends with the murky riverbeds and turbid coastal waters I often inhabit. My flanks lighten gradually, and my ventral surface is white or cream. This gradient means that prey looking up at me see my pale belly against the bright surface of the water, while prey or rivals looking down see my dark back blending with the depths. The effect is not perfect camouflage, but it breaks up my silhouette and buys me the seconds I need to close the distance on a target. My fins are broad and muscular. The first dorsal fin rises high and triangular from my back, positioned just behind my pectoral fins. It is not dramatically tall like a great white's, but it is prominent, a stabilizer that keeps me upright when I surge through currents or turn sharply in shallow water. The second dorsal fin is much smaller, a low triangle near my tail. My pectoral fins are large and paddle-shaped, extending outward and slightly downward. These are my control surfaces, my wings, allowing me to maneuver in tight spaces, bank into turns, and adjust my depth with precision. My tail, the caudal fin, is asymmetrical, the upper lobe longer than the lower, a feature called heterocircle. This shape generates lift as I swim, countering my negative buoyancy. Sharks lack the gas-filled swim bladders that bony fish use to hover in the water column, so I must keep moving or sink. My tail drives me forward with powerful side-to-side sweeps, my body flexing in smooth, efficient waves. My skin is covered in dermal denticles, tiny tooth-like scales that point backward. Under a microscope, each denticle resembles a miniature tooth, with a pulp cavity, dentine, and a hard enamel-like surface. They reduce drag as I swim, channeling water smoothly over my body. They also provide protection, making my hide tough and abrasive. A careless hand brushed backward against my flank will scrape and bleed. These denticles wear and are replaced throughout my life, a constant renewal that keeps my hydrodynamic efficiency high. My mouth is wide, curving back beneath my snout. Inside, rows of teeth line my jaws, each tooth a serrated blade designed for cutting. My upper teeth are broad and triangular, their edges finely serrated, built to saw through flesh and bone. My lower teeth are narrower, more pointed, designed to grip and pierce. Both sets lean slightly backward, ensuring that anything I bite cannot easily pull free. Behind each functional tooth, several replacement teeth wait in reserve, folded back like pages in a book. When a tooth breaks or falls out, and they do with regularity given the forces I exert and the hard structures I sometimes bite, a replacement rotates forward into position within days. Over my lifetime, I may go through thousands of teeth. This conveyor belt system means I am never without a full arsenal. My eyes are round and dark, positioned on the sides of my head, giving me a wide field of view. I lack eyelids in the mammalian sense, but I have a nictitating membrane, a translucent third eyelid that slides across the eye from the inner corner to protect it during a bite. Vision is important to me, especially in clearer water, but it is not my primary sense. My eyes contain both rod and cone cells, allowing me to see in dim light and detect contrast and motion. I do not see color as humans do, but I do not need to. I see movement, silhouettes, the flicker of a fish turning, the shadow of a mammal at the surface. More critical than vision is my sense of smell. My nostrils, or nares, are located on the underside of my snout, separated from my mouth. Water flows in through the front opening of each nostril, passes over a folded membrane packed with olfactory receptors, and exits through the rear opening. This one-way flow means I sample the water constantly as I swim. I detect amino acids, the breakdown products of protein, in concentrations measured in parts per billion. A drop of blood in a swimming pool would register. A wounded fish 100 meters upstream sends a chemical signal I cannot ignore. My brain devotes a significant portion of its processing power to scent, analyzing gradients, following concentration trails upstream, triangulating the source of a scent plume. In murky river water where visibility drops to a few centimeters, scent becomes my map and compass. I also sense the world through my lateral line, a system of fluid-filled canals running beneath the skin along both sides of my body and branching across my head. Tiny hair cells inside these canals detect pressure waves and water movements, the disturbances created by swimming prey, the wakes of other animals, the turbulence around submerged logs and rocks. This sense is sometimes called touch at a distance. It allows me to navigate in complete darkness, to track a fleeing fish even when I cannot see or smell it, to feel the current patterns that signal a channel narrowing or a sandbar ahead. Beneath my snout, scattered across my head in pores that look like tiny dots, lie the ampullae of Lorenzini. These gel-filled sacs detect weak electrical fields, the bioelectric auras generated by the muscle contractions and nerve firings of other animals. Every living thing in the water produces a faint electric signature. I sense these fields at close range, within a meter or two, using them to pinpoint prey buried in sand, to detect a motionless fish hiding in weeds, to locate the heartbeat of a mammal even when every other sense fails. This electroreception is acute and unavoidable for my prey. They cannot hide their electricity. Hearing completes my sensory array. I have no external ears, but internal structures within my head detect low-frequency vibrations traveling through the water. The thrashing of a struggling fish, the irregular splashing of an injured animal, the rumble of a boat engine, all register as sound. I am most sensitive to frequencies below 1,000 Hz, the range in which distressed prey make the most noise. A fish fighting a hook or a bird fallen into the water and flapping to stay afloat produces a beacon of sound that draws me from a considerable distance. Taken together, these senses create a perceptual world far removed from human experience. I do not navigate by sight and sound alone. I navigate by taste and smell and electric fields and pressure waves. I experience my environment as a constantly updating web of chemical trails, electrical whispers, and hydrodynamic echoes. In the turbid waters I often inhabit, this multisensory approach is not a luxury. It is a necessity. My range is the widest of any shark when measured by salinity tolerance. I am found in warm coastal waters across the globe, in tropical and subtropical latitudes, but my distribution is patchy and defined more by opportunity than strict geography. The Western Atlantic holds strong populations from the coast of Massachusetts south through the Gulf of Mexico, the Caribbean, and along the coasts of Central and South America to southern Brazil. In the Eastern Atlantic, I appear off the coasts of West Africa. The Indo-Pacific hosts me from India to Australia, throughout Southeast Asia, and across the Pacific Islands. In each of these regions, I favor river mouths, estuaries, shallow bays, and coastal lagoons, but I am equally at home in the lower reaches of rivers and sometimes venture far inland. I have been documented in the Mississippi River, though how far upstream remains debated and likely varies with river conditions. I am a regular presence in the Brisbane River in Australia, sometimes appearing kilometers upstream near populated areas. The Ganges River in India, the Zambezi River in Africa, the Amazon and its tributaries in South America, Lake Nicaragua in Central America, all have hosted me. The lake in Nicaragua is connected to the Caribbean Sea by the San Juan River, and for decades scientists debated whether bull sharks in the lake were landlocked or simply moved back and forth. Tagging studies eventually confirmed that individuals travel between the lake and the sea, though the journey involves navigating rapids and significant distances. This ability to move between marine and freshwater environments and to linger in either for extended periods sets me apart. Most sharks die in fresh water within hours. I thrive in it for weeks or months. The physiological mechanisms that allow this are complex and still being studied. In sea water, the challenge is preventing dehydration. Salt water is more concentrated than my body fluids, so water tends to diffuse out of my body, and salts tend to diffuse in. Marine sharks like my relatives address this by retaining urea and trimethylamine oxide in their blood and tissues. These compounds raise the osmotic concentration of their body fluids to match or exceed sea water, preventing water loss. The downside is that urea, which most animals excrete as a waste product, becomes a permanent resident, kept at levels that would be toxic without stabilizing compounds like trimethylamine oxide. Marine sharks also have a rectal gland, an organ near the cloaca that secretes a concentrated salt solution, expelling excess sodium and chloride. When I enter fresh water, the problem reverses. Fresh water is less concentrated than my body fluids, so water floods into my body and salts diffuse out. If I maintain the high urea levels of a full-time marine shark, I would become bloated with excess water and lose critical salts. My solution is to adjust. My kidneys, far more developed than those of most sharks, ramp up their activity, producing large volumes of dilute urine to expel excess water. At the same time, I drastically reduce urea production and allow existing urea to diffuse out or be excreted. My rectal gland reduces its output or shuts down entirely, conserving salt rather than expelling it. My gills, normally a site of passive salt loss in marine sharks, shift their function, actively absorbing salts from the surrounding water to maintain internal balance. This transition is not instantaneous. Moving from full-strength sea water to pure fresh water requires days or weeks of adjustment. During that time, I may linger in brackish zones, estuaries where salt and fresh water mix, allowing my physiology to recalibrate gradually. Juveniles adapt more quickly than adults, and females, who often enter rivers to give birth, seem particularly adept. The energy cost of this regulation is high. My metabolism must support the constant work of kidneys, gills, and rectal gland, and I must feed more frequently in fresh water to fuel these processes. But the reward is access to habitats largely free from competition with other sharks, and packed with prey that have no evolutionary history of dealing with a predator like me. Coastal waters where I spend much of my time are dynamic environments. I frequent areas with soft bottoms, mudflats, sandbars, seagrass beds, and mangrove forests. These habitats are rich in nutrients washed down from rivers or stirred up by tides and currents. Visibility is often low, the water stained brown or green by sediment and organic material. Mangroves, with their tangled roots and labyrinthine channels, provide nursery grounds for countless fish species and shelter for juvenile sharks like my younger siblings. Estuaries are mixing zones, neither fully marine nor fully fresh, where salinity fluctuates with tides, rainfall, and river discharge. I navigate these gradients with ease, moving upstream when prey is abundant, retreating to the coast when conditions change. I am capable of tolerating a wide range of temperatures, though I prefer water between 24 and 30 degrees Celsius. I have been recorded in cooler waters, particularly in temperate regions at the edges of my range, but I am less active and feed less frequently when temperatures drop. I am ectothermic, meaning my body temperature closely matches the surrounding water. I lack the regional endothermy of lamnid sharks like great whites, who can maintain their swimming muscles and core at temperatures above the ambient water. For me, warm water means faster muscle contractions, quicker digestion, and higher metabolic rates. Cold water slows me down. In tropical rivers and estuaries, temperature rarely becomes a limiting factor, but in temperate coastal waters, seasonal cooling may trigger migrations to warmer areas or deeper channels where temperatures remain more stable. My daily activity pattern is flexible, opportunistic. I have no strict schedule of dawn feeding and dusk resting. I feed when prey is available, rest when feeding opportunities diminish or after a large meal. In rivers, I often become more active during flood events when runoff washes terrestrial prey into the water and stirs up fish and invertebrates from their hiding places. In coastal areas, tides influence my movements. I may follow a rising tide into shallow bays or over submerged flats, then retreat with the falling tide to deeper channels. Moonlight affects the behavior of my prey, making some species more active and easier to detect, while others become more cautious and cryptic. I adjust accordingly, reading the rhythms of the ecosystem and inserting myself wherever vulnerability exists. Solitary behavior dominates my adult life. I do not form lasting social bonds, packs, or cooperative hunting groups. Encounters with other bull sharks are usually brief, defined by dominance displays or simply mutual avoidance. Size matters in these interactions. Larger individuals command deference. A smaller shark crossing paths with a larger one will often veer away, drop lower in the water column, or swim with exaggerated smoothness to signal non-aggression. If both are similar in size, they may circle each other briefly, each assessing the other's intent. Physical aggression between bull sharks is rare outside of mating contexts but not unheard of. I bear scars on my flanks and fins, marks left by the teeth of rivals or, perhaps, mating partners. Aggregations do occur under specific circumstances. In certain locations, multiple individuals may concentrate because resources are concentrated, not because we seek each other's company. A river channel rich in fish during a seasonal migration may host several bull sharks at once. A bay where stingrays gather to mate or feed may attract multiple individuals. These are not social groups in the sense of coordinated behavior or communication. They are overlapping home ranges, temporary clusters of individuals each pursuing their own interests. Dominance hierarchies may be loose and situational, established through displays of size and confidence rather than overt fighting. Mating introduces a different dynamic, one charged with intensity and often violence. Bull sharks are viviparous, giving birth to live young after a lengthy internal gestation. Females mature later and grow larger than males. A female reaches sexual maturity at lengths of around 160 to 220 centimeters, which corresponds to an age of 10 to 15 years, though growth rates vary with food availability and environmental conditions. Males mature earlier, at lengths of 150 to 200 centimeters, around 8 to 13 years of age. These estimates are derived from growth rings in vertebrae, a method analogous to counting tree rings, but sharks complicate this by living in environments where seasonal signals may be weak or absent. Mating occurs in late spring and summer in most populations, though timing varies with latitude and local conditions. Males locate females using scent, detecting pheromones released by receptive females. When a male encounters a female, he approaches cautiously. Females are larger and may respond aggressively. The male attempts to grasp the female's pectoral fin with his mouth, biting down to hold her in position. This bite is not gentle. Female bull sharks frequently bear fresh scars and healed wounds on their fins and flanks, evidence of mating encounters. Males have slightly longer, sharper teeth than females, an adaptation that may aid in gripping during mating. Once the male has secured a grip, he maneuvers alongside the female, aligning his body with hers. Males have paired reproductive organs called claspers, modifications of the pelvic fins that function as intermittent organs. One clasper is inserted into the female's cloaca, delivering sperm into her reproductive tract. The act is brief but forceful. The female may continue swimming throughout, attempting to dislodge the male. After copulation, the pair separates and the male may seek other females. Females do not mate again during that season. Sperm is stored internally, and fertilization occurs days or weeks after mating. Gestation lasts 10 to 11 months, one of the longer gestation periods among sharks. Embryos develop inside the female's uteri, nourished initially by a yolk sac. As the yolk is depleted, a placental connection forms, a yolk sac placenta that provides nutrients and oxygen directly from the mother's bloodstream. This is a sophisticated reproductive strategy, more similar to mammals than to egg-laying sharks. The mother does not merely carry eggs. She actively sustains growing embryos, channeling her own energy and resources into their development. A pregnant female must feed more frequently and intensively, her metabolic demands spiking as the embryos grow. Near the end of gestation, the female moves into protected shallow waters, often estuaries or the lower reaches of rivers. These nursery habitats offer abundant food for juveniles, cover in the form of vegetation or complex bottom structure, and lower densities of large predators compared to open coastal waters. Larger sharks, including adult bull sharks themselves, pose the primary threat to newborns, so females avoid areas where adults are common. The timing of birth aligns with favorable environmental conditions, often the warmer months when prey productivity is high. Birth is live and active. The female produces a litter of 1 to 13 pups, with the average around 6 to 8. Litter size correlates with female size. Larger females produce more offspring. Newborns are born tail first, emerging fully formed and immediately capable of swimming. Each pup measures 55 to 80 centimeters in length at birth, miniature replicas of the adults, complete with functional teeth and hunting instincts. There is no parental care. The moment the pups are born, they scatter and the female departs. From that instant, the pups are independent, relying on their instincts and the abundance of the nursery habitat to survive. The first weeks and months of life are the most dangerous. Juvenile bull sharks face predation from larger fish, including adult bull sharks, other shark species, and large predatory teleosts like groupers and barracudas. Birds such as herons and pelicans may take very small juveniles in shallow water. Crocodiles and alligators, where their ranges overlap with mine, are significant predators. In rivers and estuaries where crocodilians live, young bull sharks must constantly balance the need to feed in productive shallows against the risk of becoming prey themselves. Juveniles remain in nursery areas for several years, gradually expanding their range as they grow. Growth is relatively rapid in the first few years, fueled by abundant prey and the warm, productive waters of estuaries. By 2 or 3 years of age, a young bull shark may measure 100 centimeters or more, large enough to reduce the risk from most predators. As they mature, they move into deeper coastal waters and begin the adult pattern of wandering between salt and fresh water. Diet in juveniles consists primarily of small fish, crustaceans, and mollusks. I remember the early years, if memory is the right word for ingrained behavior and instinct. My siblings and I fed on mullet, anchovies, menhaden, small catfish, crabs, and shrimp. We hunted in shallow water, over mudflats and through seagrass, snapping at anything that moved and fit into our mouths. Our senses guided us: the twitch of a fish tail, the chemical signature of a wounded crab, the electrical pulse of a buried shrimp. We learned by trial and error which prey was easy to catch and which was more trouble than it was worth. As adults, my diet broadens and shifts upward in the food web. I am an apex predator in many of the habitats I occupy, and I am also an opportunist, taking whatever prey is most available or most vulnerable. Bony fish dominate my diet across most of my range. I consume species from numerous families: mullet, catfish, herrings, jacks, snook, snapper, grouper, tarpon, stingrays, and many others. I do not specialize. I am a generalist predator, capable of hunting fast swimmers, ambushing bottom dwellers, and scavenging when the opportunity arises. Stingrays and skates form a significant portion of my diet in some areas, despite their defensive barbed spines. I have been found with stingray spines embedded in my jaws, throat, and stomach lining, sometimes multiple spines in a single individual. This does not deter me. The nutritional reward of a large stingray outweighs the discomfort of a spine wound, which my body eventually walls off or expels. I hunt stingrays by pinning them to the bottom, biting down before they can bring their tail around, or by striking from behind, targeting the body and avoiding the whipping tail. Other sharks and rays, including juveniles of my own species, appear in my stomach contents. Cannibalism is not uncommon among sharks, and I am no exception. In a crowded estuary, a smaller bull shark is prey to a larger one. I have also consumed other shark species: blacktip sharks, spinner sharks, smoothhounds, hammerheads. I eat what I can catch, and I can catch a surprising variety of prey. Marine mammals enter my diet in certain regions, though they are not primary prey for most individuals. Dolphins occasionally fall victim to bull sharks, particularly young or weakened individuals. I do not often pursue healthy adult dolphins. They are fast, social, and aware, capable of mobbing and injuring a shark. But a calf separated from its pod, a dolphin sick or injured, becomes vulnerable. I detect their distress through sound and scent, and I investigate. If the opportunity presents itself, I strike. In coastal regions where terrestrial mammals wade, swim, or fall into the water, I sometimes encounter them. Dogs, pigs, cattle, horses, and even larger mammals like deer or antelope crossing rivers have been documented as prey. These encounters are opportunistic. I do not hunt land mammals as a strategy, but when one enters my domain and presents itself as prey, I respond. The electrical field of a struggling mammal, the thrashing of its limbs, the scent of stress hormones in the water, all signal opportunity. My jaws are strong enough to inflict massive trauma on a mammal, and a single bite can cause fatal bleeding. Birds resting on the water surface or diving for fish occasionally become targets. I approach from below, using countershading and the surface glare to mask my ascent, and strike upward in a sudden rush. A pelican diving for fish, a cormorant surfacing with a catch, a seabird floating on the surface. Any of these may register as prey if I am in the area and hungry. Carrion also features in my diet. A dead fish drifting downstream, a whale carcass washed into shallow water, a drowned animal, all attract me. My sense of smell leads me to these opportunities, and I feed alongside other scavengers. I am not above scavenging. A meal is a meal, and the energy saved by not hunting can be invested elsewhere. Feeding events are typically swift and violent. I am not a pursuit predator in the sense of chasing prey over long distances. My strategy is to close the distance using stealth and surprise, then strike with explosive speed. Once I detect prey, I begin a slow, careful approach, using my senses to track the target while minimizing my own profile. If the prey is near the bottom, I may swim just above the substrate, my dark dorsal surface blending with the murk. If the prey is near the surface, I ascend from below, my pale belly hidden against the bright ceiling of the water. When I am within striking range, usually a few body lengths, I accelerate. My tail beats powerfully, my body flexes, and I surge forward, jaws opening, head lifting, nictitating membrane sliding across my eyes for protection. The moment of impact is chaos. My jaws close on the target with tremendous force, my teeth slicing through flesh and bone. I shake my head violently from side to side, a behavior that uses the serrated teeth as saws, cutting through tissue and removing chunks of flesh. If the prey is small, I may swallow it whole or in a few bites. If the prey is large, I take successive bites, circling and striking repeatedly until I have consumed enough or the prey escapes or dies. My bite force has been measured and estimated at various values depending on size and jaw gape. A large adult can exert bite forces exceeding 5,000 newtons at the rear teeth, among the highest recorded for sharks of comparable size. This force, combined with sharp serrated teeth and powerful neck muscles that drive the side-to-side shaking, allows me to process prey items that would be difficult for other predators to handle. Digestion is slow, particularly after a large meal. My stomach acid is highly concentrated, capable of breaking down bone and cartilage over days. I may not feed again for a week or more after consuming a substantial prey item, during which time I rest in deeper water or in sheltered areas where current is minimal, and I can conserve energy. The pace of digestion depends on water temperature. Warmer water speeds metabolism, cooler water slows it. I am an ambush and active pursuit predator depending on circumstances. In turbid rivers, ambush dominates. I rest near the bottom or behind structure, waiting for fish to pass within range, then strike. In clearer coastal waters, I may actively patrol, covering distance in search of prey concentrations, using my senses to detect fish schools or the activity of feeding seabirds that indicate baitfish at the surface. My presence influences the behavior of prey communities. Fish and invertebrates in areas with bull sharks exhibit heightened vigilance, altered movement patterns, and shifts in habitat use. Prey species may avoid certain areas at certain times, compressing their activity into narrow windows when I am less active or less likely to be present. This is the ecological shadow of a predator, a force that shapes not only who lives and who dies, but also where and how potential prey live their lives. Predators of adult bull sharks are few. My size and power place me near the top of the food web in most of the habitats I occupy. However, I am not invulnerable. Larger sharks pose a threat, particularly great white sharks, tiger sharks, and in some regions, other large bull sharks. Great whites, where our ranges overlap in cooler temperate waters, are larger and more powerful, and they do not hesitate to attack and consume other sharks. Tiger sharks, which share much of my tropical range and also tolerate estuarine conditions, are similarly large, opportunistic, and willing to attack other sharks. Encounters between adult bull sharks and these species are probably rare, each of us avoiding confrontation when possible. But when competition for prey or space forces an interaction, size and aggression determine the outcome. Crocodilians represent a more consistent threat, especially in regions where large saltwater crocodiles or American crocodiles share my riverine habitats. These reptiles are apex predators in their own right, and they occupy similar ecological niches, patrolling the same murky waters, ambushing similar prey. Juvenile and subadult bull sharks are vulnerable to large crocodiles. Adults are more evenly matched, and confrontations may go either way depending on size, surprise, and circumstance. I avoid shallow water and basking areas where crocodiles are most common, and they likely avoid areas where I am most active. But overlap is inevitable, and neither of us gives ground easily. Humans are my most significant predator, though the relationship is complex. I am targeted by commercial and recreational fisheries in many parts of my range. My fins are valuable in the international shark fin trade. My meat is consumed fresh or dried in some regions. My liver produces oil. My skin has been used for leather. Fishing mortality has driven population declines in some areas, though the lack of comprehensive population data for many regions makes global assessments difficult. More than direct harvest, human activity affects me through habitat destruction and water quality degradation. Coastal development destroys mangroves and seagrass beds, the nursery habitats my young depend on. Dams on rivers block migration routes, fragment populations, and alter the flow and salinity regimes that define estuarine ecosystems. Pollution, agricultural runoff, industrial discharge, and sewage contamination degrade water quality, reducing oxygen levels, introducing toxins, and disrupting the prey communities that sustain me. I am capable of tolerating a range of water conditions, but I am not immune to chronic pollution. Heavy metals, pesticides, and industrial chemicals accumulate in my tissues, biomagnifying as they move up the food chain, potentially affecting my reproduction, growth, and immune function. Boat strikes injure and kill bull sharks in areas with heavy vessel traffic. Propeller wounds are common in some populations, visible as deep gashes across the dorsal surface or tail. Most individuals survive these injuries if they do not sever the spinal cord or cause catastrophic bleeding, but the scars remain, and the energy cost of healing and the increased infection risk may affect long-term survival. My reputation among humans is dominated by fear. I am considered one of the most dangerous shark species to humans, not because I am particularly aggressive by nature, but because I occupy habitats where humans frequently enter the water. River mouths, estuaries, beaches, river swimming holes, all are places where I am likely to be and where humans are likely to be. The water is often murky, limiting visibility for both me and potential prey, and my sensory system is not perfect. An arm or leg splashing at the surface, the electrical signature of a swimmer, the low-frequency vibrations of kicking feet. These signals resemble those of fish or other prey. Mistaken identity is the prevailing scientific explanation for many shark bites on humans. I investigate potential prey, and sometimes that investigation involves a bite. The consequences for the human are severe. My jaws deliver immense force, my teeth cut deep, and the shock and blood loss from a single bite can be fatal without immediate medical intervention. The majority of bull shark bites are single-strike events. I bite, realize the target is not my usual prey, and disengage. But that single bite may be enough to cause life-changing injuries. In regions where I am common and human water use is high, the risk of encounters is elevated. Australia, South Africa, the southeastern United States, Brazil, and parts of India report bull shark bites with some regularity. From my perspective, humans are not prey. They are not part of the evolutionary environment in which my hunting strategies evolved. I do not seek them out. I respond to stimuli that suggest prey. When those stimuli are ambiguous or misleading, mistakes happen. The frequency of these mistakes is low relative to the number of hours humans spend in water within my range. But each event is traumatic and reinforces a narrative of danger that overshadows the reality of my ecological role and the rarity of actual attacks. Humans also engage with me through research and conservation. Scientists have tagged and tracked bull sharks using acoustic telemetry, satellite tags, and mark recapture studies. These efforts have revealed movement patterns, site fidelity, and the extent of freshwater use. Some individuals show strong fidelity to specific rivers or estuaries, returning to the same areas year after year. Others are wanderers, traveling hundreds of kilometers along coastlines or between river systems. Males and females show different patterns in some populations, with pregnant females making more frequent and extensive use of freshwater nursery areas. Physiological studies have dissected the mechanisms of osmoregulation, revealing the interplay of kidney function, gill transport, and hormonal regulation that allows me to survive in fresh water. Genetic studies are beginning to explore population structure, asking whether bull sharks in different river systems or ocean basins are isolated populations or part of a larger pan-mictic unit. Early results suggest some genetic differentiation between regions, implying that while I am capable of long-distance movement, many individuals remain within regional areas, and gene flow between distant populations is limited. Efforts to protect bull sharks vary by region and are often incidental to broader marine conservation initiatives. Some areas have implemented fishing restrictions or bans on finning. Marine protected areas, while not typically designed specifically for bull sharks, provide refuges where I and other species are shielded from harvest. Education and outreach programs aim to reduce fear and promote coexistence, emphasizing that I am a vital part of coastal and estuarine ecosystems, not a mindless killer. Misconceptions about bull sharks are common and persistent. The idea that I am uniquely aggressive compared to other large sharks is not supported by evidence. I am opportunistic and assertive, traits that serve me well in competitive environments, but I do not attack indiscriminately. The notion that I prefer fresh water is also inaccurate. I use fresh water for specific purposes: nursery areas for females, foraging opportunities for juveniles and adults, but I remain fundamentally a marine animal, tied to the ocean for most of my life. The sensational media portrayal of bull sharks as river-dwelling monsters obscures the nuanced reality of my ecology and behavior. Another misconception is that all large sharks in rivers and estuaries are bull sharks. In some regions, other species like the sand tiger shark, the sandbar shark, or even great whites may enter brackish water or river mouths, though none penetrate as far or tolerate freshwater as long as I do. Misidentification is common, especially by observers unfamiliar with shark morphology. A blunt snout and stocky body are cited as identifying features for bull sharks, but other requiem sharks share these traits to varying degrees. Accurate identification often requires close examination or photographic evidence reviewed by experts. A typical day for me, if such a concept applies to an animal without clocks or schedules, unfolds according to hunger, tides, and the presence of prey. I begin in deeper water, a channel or bay where I have rested through the night, my metabolism ticking over slowly, conserving energy. As light filters through the water at dawn, prey species become active, moving out from shelter to feed. I sense this activity, the increase in movement, the rise in scent trails dispersing through the water. I begin to swim, slow and deliberate, my pectoral fins angled to maintain depth, my tail beating in smooth rhythm. I follow a scent trail upstream, the concentration increasing as I move, guiding me toward a school of fish congregated in shallower water where a creek enters the main channel. The turbidity is high, visibility less than a meter, but I do not rely on my eyes. I feel the pressure waves, the collective disturbance of hundreds of small fish moving as a coordinated mass. I smell the amino acids leaking from their gills and scales. I sense the crackle of their bioelectric fields overlapping and interfering like static. I approach from downstream, moving into the current, positioning myself below the school. I ascend slowly, closing the distance. When I am beneath them, I accelerate, jaws opening, head angling upward, and I drive into the school. Fish scatter in all directions, a chaos of movement and panic. I snap my jaws closed on one, feel the crunch of bones, swallow, and wheel around for another pass. The school disperses, fleeing into deeper water or hugging the bottom, and I pursue the nearest cluster, managing to take one more fish before they escape my reach. I settle to the bottom, letting the fish I have swallowed slide down my throat and into my stomach. It is not enough to satisfy me fully, but it takes the edge off hunger. I resume patrolling, moving along the channel, pausing near structure, logs, and undercut banks where larger fish might hide. Hours pass. The sun climbs higher and the water temperature rises slightly. Tides shift and I feel the current slow, then reverse. I move with the tide, letting it carry me upstream into shallower water. In the shallows, I encounter a ray partially buried in sand. My ampullae of Lorenzini detect its electric field, a faint but distinct signal beneath the substrate. I approach carefully, position myself, and strike downward, pinning the ray with my jaws before it can flee or deploy its spine effectively. It thrashes and I bite down harder, my teeth cutting through cartilage and muscle. I shake my head, tearing away a large piece of tissue. The ray weakens, its movement slowing. I feed methodically, taking successive bites until only scattered fragments remain. This meal is substantial. I retreat to deeper water, seeking a place with minimal current where I can rest. I settle near the bottom, my body angled slightly nose up into the faint flow, and I allow my digestion to begin. My stomach churns, acid breaking down tissue, my metabolism ramping up to process the nutrients. I remain here for hours, occasionally adjusting position but largely motionless, conserving energy. Other fish pass nearby, but I do not pursue them. My hunger is sated. As the day wanes and evening approaches, the tide begins to fall. I follow it back downstream toward the bay, moving through water that grows progressively saltier. My physiology begins the slow adjustment, my rectal gland activating to expel excess salt, my kidneys reducing urine output. I pass through the estuary, where mangroves line the banks and channels branch in all directions, and I emerge into the open bay where deeper water and stronger currents prevail. The cycle will repeat tomorrow, and the day after, and the day after that, each day shaped by the availability of prey, the state of my hunger, the rhythm of the tides, and the chance encounters that define a predator's life. I am part of the system, both shaping it and shaped by it, a thread in the web of energy and nutrients that flows through coastal and estuarine waters. My ecological role extends beyond the direct predation I inflict. I influence the distribution and behavior of prey, creating landscapes of fear that structure ecosystems in subtle ways. Prey that must remain vigilant, that must flee at the slightest disturbance, that must avoid certain areas or restrict activity to certain times, pay costs in energy and opportunity. These costs ripple through the food web, affecting growth rates, reproductive success, and population dynamics. My presence may indirectly benefit some species by suppressing their competitors or predators, a phenomenon ecologists call a trophic cascade. I also serve as a host for parasites, some highly specialized to sharks. Copepods cling to my skin and gills, feeding on mucus and tissue. Tapeworms inhabit my digestive tract, absorbing nutrients from my meals. Isopods attach to my fins. These parasites are rarely fatal, but heavy infestations can weaken me, reducing my swimming efficiency and increasing energy demands. Some parasites have complex life cycles involving multiple hosts, using me as the final stage where they mature and reproduce. When I defecate, I release eggs or larvae into the water, dispersing them to new locations and continuing the cycle. I am also susceptible to disease, though less is known about pathogens in wild sharks compared to terrestrial mammals or captive animals. Bacterial infections can take hold in wounds, particularly deep bites from rivals or injuries from fishing gear. Viral and fungal pathogens likely exist but are poorly documented. My immune system, primitive by mammalian standards, relies on innate defenses, antimicrobial compounds in my skin mucus, white blood cells that engulf pathogens, and inflammatory responses that wall off infections. I lack the adaptive immune system with memory cells that allows mammals to develop long-term immunity, but my innate system is effective enough for survival in the pathogen-rich aquatic environment. My lifespan is estimated at 25 to 30 years or more in the wild, though precise aging is difficult, and some individuals may live longer. Mortality is highest in the first year of life, when predation and starvation claim the majority of each cohort. Survivors face declining mortality risk as they grow, reaching a relatively stable adult phase where annual survival is high but not absolute. Senescence, the process of aging and declining function, is poorly studied in sharks. I likely experience some decline in swimming performance, sensory acuity, and healing capacity as I age. But whether this translates to reduced foraging success or increased vulnerability to predators is unknown. Females invest heavily in reproduction, each pregnancy requiring months of elevated energy intake and culminating in the production of multiple large offspring. This investment takes a toll. Females may not reproduce every year, instead following a biennial or triennial cycle, allowing time to recover body condition between pregnancies. Males, with their lower reproductive investment per mating event, may be capable of mating annually, though competition for access to females means that not all males reproduce each year. Reproductive success is likely skewed, with larger, more dominant males siring a disproportionate share of offspring. The genetic diversity of bull shark populations is maintained by this reproductive strategy, but it is also influenced by habitat fragmentation and population size. Small, isolated populations face the risk of inbreeding and loss of genetic variation, which can reduce fitness and adaptability. Connectivity between populations, mediated by the movement of individuals along coastlines or between river systems, is critical for maintaining genetic health. Conservation strategies that protect migration corridors and reduce barriers to movement, such as removing obsolete dams or ensuring minimum flow regimes in rivers, support genetic connectivity and population resilience. Climate change poses emerging threats. Rising ocean temperatures may expand my range poleward, but they also alter the productivity and structure of coastal ecosystems, potentially disrupting prey availability. Ocean acidification, the absorption of atmospheric carbon dioxide by seawater, lowers pH and affects the physiology of many marine organisms, particularly those with calcium carbonate structures. I am not directly affected by acidification since my skeleton is cartilage, but the prey species I depend on, many of which have bones or shells, may suffer, indirectly impacting me. Sea level rise and increased storm intensity affect estuarine and mangrove habitats, the nurseries my young require. Changes in river flow regimes, driven by altered precipitation patterns and increased water extraction for human use, affect salinity gradients and the availability of brackish and freshwater habitats. Human responses to climate change, such as coastal development to protect infrastructure or alterations to river systems to manage water supply and flood risk, may further fragment and degrade the habitats I depend on. The cumulative impacts of these changes are difficult to predict, but they represent significant challenges for a species that depends on the interface between land and sea, fresh water and salt. My interactions with other shark species are shaped by competition and niche partitioning. In coastal waters, I overlap with sandbar sharks, blacktip sharks, spinner sharks, lemon sharks, and others. Each species has slightly different preferences in depth, salinity, temperature, and prey, reducing direct competition. Sandbar sharks tend to favor slightly cooler water and deeper habitats. Blacktip sharks are more surface-oriented and more common on coral reefs. Lemon sharks use similar nursery habitats but may feed more heavily on benthic invertebrates. I occupy a generalist niche, overlapping with many species but not completely redundant with any. Tiger sharks, my closest ecological counterpart in terms of size, generalist diet, and tolerance of estuarine conditions, represent the most direct competition. Where we coexist, partitioning likely occurs along subtle gradients of depth, turbidity, or prey preference. Tiger sharks may forage more extensively in open water and on reefs, while I focus more on rivers and turbid estuaries. But the overlap is real, and in areas of high productivity, both species may be present and competing for the same prey resources. Great hammerhead sharks also share portions of my range and diet, particularly in their preference for stingrays. Hammerheads use their unique head shape to detect and pin rays to the bottom, a strategy different from mine, but targeting the same prey. Competition between hammerheads and bull sharks is probably more about access to productive areas than direct interference, each species exploiting rays in slightly different ways or at different times. Within the broader community of coastal predators, I am part of a guild that includes large teleost fish like groupers, barracudas, and tarpon, as well as marine mammals like dolphins and, in some areas, seals. These predators overlap in diet but differ in hunting strategies and sensory modalities. Dolphins, with their echolocation and social cooperation, can locate and capture prey in ways I cannot. Groupers, with their ability to hover motionless and ambush from cover, exploit opportunities I might miss. Barracudas, with their speed and streamlined bodies, pursue fast-swimming prey in open water more effectively than I do. The coexistence of all these predators suggests that the prey base is diverse and abundant enough to support multiple guilds, each taking its share. I sometimes scavenge on the kills of other predators or feed on prey injured by fishing activity. A fish hooked by an angler and fighting on the line is a distress signal broadcast across the water, and I may home in on it, biting the fish or the line, or, in rare cases, the angler's catch as it is being reeled in. This behavior brings me into direct conflict with fishermen, who view me as a nuisance that steals their catch or damages their gear. From my perspective, the struggling fish is simply an easy meal, the work of capture already done. My relationship with cleaner organisms is less studied than in other large fish, but it is likely that I benefit from interactions with cleaner wrasses, gobies, or shrimp in certain habitats. These small animals remove parasites and dead tissue from larger fish, providing a service that improves health and comfort. Whether I seek out cleaning stations as some reef sharks do, or whether cleaners opportunistically pick at me when I rest in suitable areas, is not well documented. Given the time I spend in murky, low-visibility environments, cleaning interactions are probably less frequent and less ritualized than in clear water reef systems. My contribution to nutrient cycling is significant. When I feed in one location and defecate in another, I transport nutrients across the landscape. Feeding in a river and defecating in the ocean, or vice versa, moves nitrogen, phosphorus, and other elements between ecosystems. My urine and feces fertilize the water, supporting the growth of phytoplankton and bacteria, the base of the aquatic food web. My carcass, when I eventually die, provides a resource pulse for scavengers and decomposers, returning all the nutrients I accumulated over my lifetime back into the ecosystem. Death may come from many sources. Disease, injury, starvation, predation, and old age all take their toll. A significant portion of adult mortality in many populations is now attributable to human activity, fishing, bycatch, boat strikes, and entanglement in nets or lines. A bull shark caught in a gill net and unable to escape will drown, suffocating as water flow over the gills ceases. A shark hooked on a long line may be hauled aboard and killed for fins and meat, or cut loose with the hook still embedded, leading to chronic injury and reduced feeding ability. When death occurs in the water, my body sinks or drifts, depending on size and buoyancy. Scavengers respond quickly. Other sharks, including bull sharks, may feed on the carcass. Crabs, amphipods, and other invertebrates swarm over it, stripping away soft tissue. Bacteria and fungi break down what remains, releasing nutrients into the water and sediment. Within weeks or months, little remains but cartilage and teeth, and even these may be scavenged or buried. The teeth, however, endure. Fossilized shark teeth are among the most common vertebrate fossils, a testament to the durability of enameloid and the sheer number of teeth a shark produces over a lifetime. My ancestors left behind teeth that tell the story of sharks over hundreds of millions of years, a record of evolution, diversification, and adaptation that stretches back to the Devonian period, long before the dinosaurs. The fossil record shows that requiem sharks, my family, have existed for tens of millions of years, with modern forms emerging in the Miocene. Bull sharks, as a distinct species, are probably much younger, emerging in the Pliocene or Pleistocene. Tracing the exact lineage is challenging because shark skeletons, being cartilage, rarely fossilize intact. Teeth and vertebrae are the primary fossils, and while teeth reveal much about diet and ecology, they tell less about overall body form and behavior. The evolutionary innovation that allows me to tolerate fresh water is unique among living sharks, but may have precedence in extinct lineages. Some ancient sharks likely invaded freshwater systems, and a few modern species besides myself, such as the river sharks of the genus Glyphides, also tolerate low salinity. These species are rare and poorly known, inhabiting remote tropical rivers in Australia and Southeast Asia. They are even more specialized for freshwater life than I am, with some populations apparently resident in fresh water year-round. The evolution of freshwater tolerance appears to have occurred multiple times independently in sharks, suggesting that the physiological barriers, while significant, are not insurmountable given sufficient evolutionary time and selective pressure. The selective pressures that drove the evolution of my freshwater tolerance likely involved competition and predation in the marine environment and the availability of unexploited resources in rivers. Estuaries and rivers, particularly in the tropics, are extraordinarily productive, supporting high densities of fish and invertebrates. A shark capable of accessing these resources while avoiding competition from purely marine predators would have a significant advantage. The costs, the energetic demands of osmoregulation, and the challenges of navigating confined, variable habitats were outweighed by the benefits of access to abundant prey and safe nursery areas. Today, I am a living representative of that evolutionary experiment, a marine predator that ventures into fresh water, a bridge between two worlds. My physiology is a compromise, efficient in neither pure salt nor pure fresh water, but functional in both and optimized for the gradient in between. My behavior is flexible, allowing me to exploit opportunities across a range of salinities and habitats. My ecology is complex, intertwined with both marine and freshwater systems, making me a key species in the coastal and estuarine ecosystems I inhabit. Understanding my role in these ecosystems requires understanding the ecosystems themselves. Estuaries are among the most productive ecosystems on Earth, rivaling tropical rainforests and coral reefs in terms of biomass production. They receive nutrients from both land, via rivers carrying runoff, and sea, via tides carrying plankton and detritus. The mixing of salt and fresh water creates a unique chemical environment that supports specialized communities of plants and animals. Mangroves and salt marshes fringe estuaries, their roots stabilizing sediments, their leaves producing organic matter that fuels the food web. Seagrass beds carpet shallow areas, providing habitat and food for invertebrates and fish. Mudflats and sandbars shift with tides and storms, constantly reshaping the landscape. This dynamism is both opportunity and challenge for me. I must navigate a constantly changing environment, adapting to shifts in salinity, temperature, water level, and prey availability. My wide physiological tolerances and behavioral flexibility allow me to thrive in this variability. I am not confined to a narrow niche, but instead occupy a broad one, positioning me as a generalist predator capable of persisting through environmental fluctuations that might eliminate more specialized species. My presence in an estuary influences the entire community. By preying on fish and invertebrates, I exert top-down control, limiting prey populations and altering their behavior. This can have cascading effects. If I reduce the abundance of a fish species that feeds on invertebrates, those invertebrate populations may increase, which in turn affects the algae or detritus they consume. The strength and direction of these cascades depend on the structure of the food web, the presence of other predators, and environmental factors like nutrient availability and disturbance regimes. Empirical studies of trophic cascades involving sharks are challenging because sharks are difficult to manipulate experimentally. Researchers have used natural experiments, comparing areas with and without sharks, or historical comparisons, examining ecosystems before and after shark population declines. These studies suggest that sharks, including bull sharks, play important roles in structuring communities, but the effects are context-dependent and often subtle. The loss of sharks does not lead to immediate, dramatic shifts in most cases, but over time, ecosystems without sharks may show altered species compositions, different size structures in prey populations, and changes in habitat use patterns. One well-documented consequence of shark declines is the phenomenon called mesopredator release. When top predators are removed, mid-level predators, mesopredators like smaller sharks, rays, and large fish, increase in abundance. These mesopredators may then exert increased pressure on their own prey, potentially leading to declines in commercially or ecologically important species. For example, in some regions, declines in large sharks have been linked to increases in cow-nose rays, which in turn have been implicated in declines of shellfish populations. The exact relationships are debated and complex, but the general principle is supported. Removing top predators disrupts the balance of the system. If bull sharks decline in estuaries and coastal waters, the consequences would likely include changes in fish community structure, altered behavior and distribution of prey species, and potential increases in mid-level predators. Juveniles of my species would also be affected, since adult bull sharks are predators of juveniles. A reduction in adult abundance could lead to increased juvenile survival, a compensatory response, or it could reduce the selective pressure that shapes juvenile behavior and habitat use, leading to longer-term evolutionary changes. Conservation of bull sharks requires protecting not just the sharks themselves, but also the habitats they depend on. Estuarine and riverine nursery areas must be maintained in a condition that supports high prey productivity and provides cover for juveniles. This means protecting mangroves from coastal development, maintaining water quality through pollution control and sustainable land use practices, and managing river flow regimes to sustain the salinity gradients that define estuarine ecosystems. Fishing mortality must be managed to prevent overharvest, which requires monitoring catch, enforcing regulations, and addressing illegal and unreported fishing. Public perception is a significant barrier to conservation. Fear of sharks drives opposition to protective measures and fuels demand for shark culling programs in some regions. Shark nets and drumlins, installed on beaches to reduce the risk of shark bites, kill large numbers of sharks, including bull sharks, as well as other marine animals like dolphins, turtles, and rays. These programs are controversial, with conservationists arguing that they are ineffective and ecologically damaging, while proponents argue that they provide psychological comfort to beachgoers and may reduce risk in heavily used areas. Alternative approaches to reducing human-shark interactions include education, promoting awareness of shark behavior, and when and where encounters are most likely, surveillance, using drones or spotter planes to detect sharks near beaches and alert swimmers, and non-lethal deterrents, such as personal electronic devices that emit electrical fields to deter sharks. The effectiveness of these alternatives varies, and no single approach eliminates risk entirely. The challenge is to find solutions that balance human safety, conservation, and the reality that sharks are a natural part of coastal ecosystems and have as much right to inhabit these spaces as any other species. My story is not just one of survival and predation, but also one of connection. I connect river and ocean, fresh water and salt, the terrestrial and marine realms. I move nutrients and energy across boundaries that most organisms cannot cross. I embody the fluidity of nature, the idea that ecosystems are not isolated units but interconnected components of a larger whole. The health of the river affects the estuary, which affects the coast, which affects the open ocean. I move through all these zones, and my fate is tied to the fate of each. As human activity continues to alter coastlines, rivers, and oceans, my future remains uncertain. Some populations are stable or even increasing in areas with strong management and protection. Other populations are declining, their habitats degraded, their numbers reduced by overfishing and bycatch. The global picture is incomplete, hindered by the difficulty of surveying wide-ranging marine species and the lack of long-term data for many regions. What is clear is that I am adaptable. I have survived millions of years of environmental change, ice ages, sea level fluctuations, and shifts in climate and ocean chemistry. I have adapted to exploit new opportunities like the invasion of freshwater habitats, but adaptability has limits. Rapid, human-driven changes, habitat destruction, overfishing, pollution, and climate change are happening at a pace that evolution cannot match. My ability to survive depends not just on my biology, but on the choices humans make about how to manage the planet's resources and ecosystems. I continue my existence as I always have, patrolling the murky waters of rivers and coasts, hunting, resting, mating, giving birth. I do not reflect on the past or worry about the future. I live in the present, responding to hunger and opportunity, shaped by instincts honed over countless generations. I am a bull shark, Carcharhinus lucas, a predator of two worlds, and this is my life. The early morning light filters through the water column in thin, slanting rays, diffused by the suspended sediment that gives the estuary its characteristic brown-green hue. I have spent the night in a deep channel, my body angled into the gentle current, my metabolism slow, conserving energy. As the sun rises and water temperature begins to climb, my activity level increases. I am a creature of temperature, my biochemistry accelerating with warmth, slowing with cold. A rise of just a few degrees sharpens my senses, quickens my muscle contractions, makes me more alert and more dangerous. I begin to move, my tail sweeping in lazy arcs, my pectoral fins adjusting angle to maintain position just above the bottom. The channel is perhaps 6 meters deep here, the bottom a mix of mud and shell fragments, the water opaque beyond a meter or two. I do not need to see far. My other senses create a three-dimensional map of my surroundings, updating in real time as I move. A school of mullet passes overhead, their electrical fields flickering in my ampullae, their bodies displacing water in patterns my lateral line reads as size, number, and direction. They are not worth pursuing yet. They are moving fast, alert, and I am not positioned for an ambush. I file the information away, a mental note that this channel holds mullet, and I continue upstream. The tide is beginning to rise, pushing salt water inland, and I ride the leading edge of this saline intrusion. The boundary between salt and fresh is not sharp but gradual, a mixing zone where the two mingle in swirling eddies and layered currents. I am comfortable in this zone, my physiology tuned to handle the gradual shifts in salinity. I have been in this estuary for several weeks, moving up and down with the tides, feeding opportunistically, and I have encountered several other bull sharks during this time. One was larger than me, a female, possibly pregnant. We passed with minimal interaction, her dominance clear in her size and the confidence of her movements. Another was smaller, a young male, and he gave me a wide berth, veering into shallower water to avoid a close encounter. Ahead, the channel narrows and the current strengthens. I feel the resistance against my body and adjust my swimming effort, increasing tail beat frequency to maintain speed. The banks here are lined with mangroves, their roots arching into the water like the ribs of some ancient beast. The roots provide structure, habitat for small fish, crabs, and oysters. I swim slowly past, my senses probing the tangle of roots. A small snapper darts out, fleeing my approach. I snap at it, my jaws closing a fraction too slow, and it vanishes into the safety of the roots. I do not pursue. The effort of navigating the roots outweighs the reward of a single small fish. Further upstream, the water shallows to 3 meters, then 2. Submerged vegetation appears, ribbons of eelgrass swaying in the current. I slow, moving cautiously, my dorsal fin occasionally breaking the surface. In water this shallow, I am conspicuous, and while I am the predator here, I do not take visibility lightly. Large wading birds, herons, and egrets stand motionless in the shallows, their keen eyes scanning for movement. They are not a threat to me, but their alarm calls can alert my prey, making hunting more difficult. A disturbance upstream catches my attention, a sudden splash, the low-frequency thud of something striking the water. My hearing, tuned to these sounds, processes the signal, irregular, chaotic, not the clean splash of a diving bird but the flailing of an animal in distress. I accelerate, my body flexing with more urgency, my senses narrowing focus on the source of the sound. As I close the distance, the scent hits me, a mammal, fresh blood, adrenaline. My predatory circuits activate fully, my pupils dilating, my metabolism ramping up, preparing for the explosive effort of a strike. I see the shape at the surface, a dark mass thrashing, limbs kicking. A dog, a domestic animal, has fallen or jumped into the water and is struggling to swim in the current. It is not far from shore, but the current is pulling it away from the bank, out into deeper water. The dog's panic broadcasts in every sensory modality I possess. The splashing sound, the chemical signature of stress, the electrical storm of its nervous system firing at maximum intensity. To me, it is indistinguishable from distressed prey. I approach from below and behind, ascending as I close the distance. My vision is poor in this turbid water, but I do not need it. I feel the dog's kicks through my lateral line, I smell its scent, I sense the electric field of its beating heart. At the last moment, I open my jaws wide, my nictitating membranes slide across my eyes, and I strike upward, my teeth closing on a hind leg. The impact is violent, my momentum carrying both of us briefly out of the water. The dog yelps, a sound I barely register, and thrashes wildly. I shake my head, the instinct to saw through tissue overwhelming, and I feel flesh and bone part. The dog's struggles weaken. I release, pulling back, circling. Blood pours into the water, a dense cloud that my senses track easily. The dog is still alive, still moving, but it is grievously injured, its leg severed at the knee. It tries to swim, but it is weakening rapidly, shock and blood loss taking hold. I make another pass, this time striking the body, my jaws closing on the torso. The force of the bite crushes ribs and the dog goes limp. I do not kill for sport or out of malice. I kill because this is what I do. This is my role in the ecosystem, to remove the weak, the injured, the unlucky. The dog's entry into the water was an accident, a mistake, but nature does not forgive mistakes. I feed methodically, taking large bites, consuming muscle and organ tissue. The dog is not large, and within minutes there is little left but scattered fragments of bone and fur drifting in the current. I am sated, my stomach full, and I retreat downstream back toward deeper water. The event will likely be discovered. The dog's owner will search, perhaps find remains, perhaps blame me or a crocodile. To me, it was simply a meal, an opportunity that presented itself and was taken. This is the reality of my existence, stripped of sentimentality. I am an opportunist, a predator without conscience, driven by hunger and instinct. I do not distinguish between a dog and a fish except in the mechanics of capture and consumption. Both are potential prey if they enter my domain and present themselves as vulnerable. The human world, with its categories of pet and wild animal, its moral distinctions between acceptable and unacceptable predation, does not penetrate my awareness. I am a shark, and I do what sharks do. The weeks pass and the estuary enters a new phase. Heavy rains upstream swell the river, increasing freshwater discharge, and the salinity in the estuary drops. The mixing zone shifts seaward, and I move with it, following the brackish water I prefer. In the swollen river, visibility is even worse, the water laden with sediment and organic debris washed down from the watershed. Logs and branches tumble past, carried by the current, and I navigate carefully, avoiding collisions. The flood brings new prey. Terrestrial insects, worms, and small animals are swept into the water, and fish gather to feed on this bounty. I find a concentration of catfish in a side channel, dozens of them, feeding on drowned insects and detritus. Catfish are armored with spines and tough skin, but I have learned to handle them. I approach from the side, biting behind the head, avoiding the pectoral and dorsal spines. I catch several, each one a substantial meal, high in fat and protein. My stomach fills and I rest, letting digestion proceed. During this rest period, another bull shark, a large male, enters the channel. He is slightly smaller than me, but not by much, and he is clearly hungry, his movements quick and searching. I watch him from my position near the bottom, my body still, conserving energy. He detects the catfish, attacks, and feeds. I do not challenge him. There is enough prey for both of us, and the energy cost of a fight outweighs any benefit. We feed in proximity, perhaps 20 meters apart, each aware of the other but not interacting. This uneasy coexistence is typical. Bull sharks are not social, but we are not strictly territorial either. Territoriality requires defense of a defined area, expelling intruders and maintaining exclusive access to resources. I do not do this. I occupy a home range, an area I know well and move through regularly, but I tolerate other individuals as long as resources are not severely limited. When food is scarce, competition intensifies and interactions become more aggressive. When food is abundant, as during this flood, multiple individuals can coexist with minimal conflict. As the flood recedes, salinity begins to rise again and I adjust my position, moving upstream once more. I have been in this system for nearly 2 months and I am now intimately familiar with its geography. The channels, the shallows, the mangrove forests, the sandbars, the deep holes. This familiarity is a significant advantage. I know where prey congregates, where currents create ambush opportunities, where I can rest undisturbed. A stranger entering this system lacks this knowledge and must spend time exploring, learning, taking risks. One evening, as the sun sets and light fades, I detect a new scent, a female bull shark, receptive, ready to mate. Her pheromones are unmistakable, a chemical signal that triggers a shift in my behavior. My feeding drive diminishes, replaced by reproductive drive. I follow the scent trail, swimming upstream, the concentration increasing. I pass through areas I know well, my attention narrowed to the pursuit of this female. I find her in a deeper pool, circling slowly. She is large, longer than me by perhaps 20 centimeters, her body robust, her movements deliberate. I approach cautiously, signaling my interest. She does not flee, but she does not stop moving either. I swim alongside her, matching her pace, and she tolerates my presence. I move closer, attempting to grasp her pectoral fin with my jaws. She accelerates, pulling away, testing my persistence. I pursue, closing the distance again, and this time I manage to bite down on her fin, holding firm. She struggles, thrashing, trying to dislodge me. My teeth cut into her fin, drawing blood, but I do not release. This is part of the mating ritual, brutal by human standards, but normal for us. I maneuver my body alongside hers, bending to align our cloaky. She continues to resist, swimming hard, but I maintain my grip. I insert one clasper, delivering sperm, the act brief and forceful. Fertilization occurs internally, and the female stores the sperm, using it over the coming days or weeks to fertilize her eggs. After copulation, I release her and she accelerates away, disappearing into the murky water. I do not follow. My drive is sated, at least for now. I bear minor wounds from the encounter, scrapes from her rough skin, and perhaps a nip she managed to inflict during her struggles. These will heal in time, adding to the collection of scars I carry. The female will now enter a long gestation period, nearly a year, during which the embryos develop inside her. She will need to feed intensively to support this process, and she will likely move into fresh water when the time comes to give birth, seeking the safety of nursery areas for her pups. I will not be part of that process. My role ends with copulation. I will never meet my offspring, never protect or provision them. They will be born independent, miniature predators, and they will survive or die based on their own abilities and the conditions they encounter. This detachment, this absence of parental care, is typical for sharks and many other fish. It contrasts sharply with the extended parental investment seen in mammals and birds, where parents feed, protect, and teach their young. In my world, investment is front-loaded. The female invests energy and resources into producing large, well-developed offspring, and then she releases them into an environment where survival is uncertain. The strategy relies on numbers and the quality of the nursery habitat. Some pups will die, many will die, but some will survive, and those survivors carry forward the genetic legacy. This reproductive strategy shapes my life history. I mature slowly, reaching reproductive age only after a decade or more. I produce relatively few offspring compared to species that lay thousands of eggs. But each offspring is large, well-formed, and equipped with the sensory and locomotor abilities needed to survive. This is a bet on quality over quantity, a strategy suited to environments where juvenile survival rates, while low, are not catastrophically low, and where the long lifespan of adults allows for multiple reproductive events over a lifetime. The seasons turn, though in the tropics, the distinction between summer and winter is subtle, marked more by rainfall and river discharge than by temperature. I remain in the estuary through the dry season, a period of reduced freshwater input and increasing salinity. The estuary shrinks, the salinity gradients compress, and the system becomes more marine in character. Some fish species that favor fresh water retreat upstream, concentrating in river pools and smaller tributaries. I follow them part of the way, but I do not venture into the purest freshwater reaches. There are limits to my tolerance, thresholds beyond which the energetic cost of osmoregulation outweighs the benefits of access to prey. During the dry season, competition increases. With less habitat and more compressed prey populations, I encounter other predators more frequently. A large tiger shark appears in the lower estuary, a transient visitor drawn by the concentration of prey. The tiger shark is larger than me, more heavily built, and I avoid her. We pass at a distance, each aware of the other, each respectful of the other's power. She feeds on sea turtles and rays, her jaws strong enough to crush shells, her diet broader even than mine. She does not linger, moving on after a few days, and I reclaim the areas she temporarily displaced me from. A young saltwater crocodile takes up residence in a section of the estuary I often frequent. This crocodile is perhaps 3 meters long, large enough to be a serious competitor but not yet fully grown. Crocodiles are ambush predators, relying on stealth and explosive strikes to capture prey at the water's edge or just beneath the surface. The crocodile and I hunt in overlapping spaces but with different tactics. The crocodile waits, motionless, camouflaged among the mangrove roots, while I patrol actively, covering ground, searching for scent trails and electrical signals. We occasionally cross paths, and each time there is a moment of assessment. I am more mobile, able to flee into deeper water if necessary. The crocodile is more dangerous at close range, its bite force exceeding even mine. Neither of us seeks a confrontation, and we maintain a wary distance. This coexistence is pragmatic, not peaceful. If I were smaller, the crocodile would likely attack me. If the crocodile were smaller, I might attempt to prey on it. At our current sizes, we are too evenly matched, and the risk of injury discourages aggression. This is the calculus of predation, the probability of success, the expected reward, the potential cost. When the cost exceeds the benefit, predators back down, even apex predators like us. The wet season returns, the rains resume, and the cycle begins again. Freshwater floods the estuary, salinity drops, and the system expands. I move upstream, following the brackish zone, feeding on the influx of prey that accompanies the flood. Another year has passed, another cycle completed. I have survived another year, grown slightly, added more scars to my hide, accumulated more experience in the art of predation. I am now approaching my peak years, the period of maximum size and strength, the years when I am the most formidable predator in my range. But time moves differently for me than for humans. A year is not a unit I mark or measure. It is simply a passage of seasons, a repetition of environmental cues, a series of feeding opportunities, threats avoided, and encounters with rivals and mates. My sense of time is immediate and cyclical, tied to tides, temperature, hunger, and reproductive condition. I do not plan for the future or reminisce about the past. I exist in the now, responding to the world as it presents itself, moment by moment. In this eternal present, I am both ancient and new, carrying within me the accumulated adaptations of millions of years of shark evolution, while experiencing each day as a fresh set of challenges and opportunities. I am Carcharhinus lucas, a name that means nothing to me but encapsulates, for humans, my identity and my place in the tree of life. I am a bridge between worlds, a predator that defies easy categorization, a survivor in a changing world. And I continue as I always have, moving through water, hunting, resting, reproducing, living the life that evolution has shaped for me.